Discussion Board Number 2, Zoology 142, Lecture, Spring 2015 by Britney F. Topic: Describe the feedback system that controls the respiratory rate. Breathing patterns are determined by carbon dioxide and oxygen concentrations in the blood. Chemoreceptors detect such chemical changes. Central chemoreceptors account for 70% of the carbon dioxide responses. They are located in the brainstem. Peripheral chemoreceptors contain cells sensitive to oxygen levels. Carotid bodies, shown here with the two arrows above, are located in the carotid artery. Aortic bodies are located in the aortic arch. Carotid bodies are the main oxygen sensors. The respiratory center, also known as the control center, responds to the changes and sends signals towards the effectors, known as the external intercostal muscles and the diaphragm. The pontine respiratory system, located in the pons, consists of clustered neurons called the pontine respiratory group. This group transmits impulses to the VRG in the medulla that modifies the breathing rhythms. The medullary respiratory system has clustered neurons in two areas. Shown in the blue, the VRG, the ventral respiratory group, and in the green to the right, the DRG, the dorsal respiratory group. The ventral respiratory group is a rhythm-generating center. It consists of neurons that fire either during expiration or inspiration to send impulses to excite the diaphragm and external intercostal muscles to move at the desired rhythm. The dorsal respiratory group integrates input from the peripheral stretch and chemoreceptors and relays the information to the ventral respiratory group. Carbon dioxide concentration increases and oxygen levels decrease. Central and peripheral chemoreceptors, mainly the aortic bodies, respond to the increase in carbon dioxide levels. This then triggers a negative feedback mechanism that increases respiratory rate and depth. The control center, known as the respiratory center, then sends additional signals to contract the diaphragm and the external intercostal muscles at a faster pace. This means more oxygen intake and expels more carbon dioxide until levels are then restored to a homeostatic state. When carbon dioxide decreases and oxygen increases, chemoreceptors, mainly the carotid bodies, respond to the increase in oxygen levels, which then triggers a feedback that decreases the respiratory rate and depth. The respiratory center sends fewer signals to the diaphragm and external intercostal muscles to contract at a slower pace. This means more carbon dioxide and less oxygen intake until levels are then restored. Homeostatic imbalances result in abnormal readings in the respiratory rate and depth. Chronic obstructive pulmonary diseases, COPD, such as emphysema and chronic bronchitis, exemplify this. Emphysema is the destruction of alveolar walls and a breakdown of elasticity in the connective tissue of the lungs. Chronic bronchitis is a chronic mucus production and cough. These may result in hypertension and a process to then restore homeostasis. During hypertension, an increased rate and depth of breathing occurs and results in a drop of carbon dioxide concentrations. In order to restore the levels of carbon dioxide and oxygen, respiration becomes slow and shallow.